Thank you very much, Ili. Good afternoon, everyone. Before introducing myself, even though Ili has done it already, I'd like to thank the organizers for establishing such a great community and giving me the opportunity to present my work to all of you. I am Davide Salzano, as Ili said, and I am a postdoctoral researcher at Scuola Superiore Meridionale in Naples. My research focuses on the development of biomolecular controllers to regulate gene expression in microbial consortia. Today, I will be presenting you a work on the in vivo distributed multicellular control of gene expression in synthetic microbial consortia, which was done in collaboration with the Savory, Marucci, and Grierson laboratories at the University of Bristol. So let me start with a brief introduction to why it is important to control biomolecular systems. As you all know, synthetic biology aims at embedding cells with novel and useful functionalities, enhancing their capabilities for a wide range of applications, ranging from chemical and pharmaceutical industries to sustainable fuel production and also agriculture. In most applications, it is necessary to assemble different modules that need to work together to achieve a common goal. However, due to factors such as stochasticity, retroactivity, metabolic burden, often when you mix together different modules, the outcome is not what you expect. So you expect an ideal response like this one, and you get something that is very noisy and doesn't behave exactly as you expect. That's why in the past decade, scientists have tried to improve the robustness and the reliability of synthetic modules by designing embedded biomolecular controllers that can guarantee reliable, robust, and tunable control of gene expression, so that the ideal response matches exactly with the outcome of the biological process that we want to control. Different implementations have been proposed of embedded controllers during the last years, and among the solutions proposed, the realization of biomolecular PID controllers has achieved a resounding success. It can enable the transfer of the reliability and robustness guaranteed by classical PID controllers in mechanical and electrical systems to synthetically engineered cells. In 2019, in this pioneering work by Chevalier and others, a first design and in silico validation of an embedded biomolecular PID controller was proposed and analyzed in silico. From this seed, different implementations have been proposed using different components and different mechanisms to realize a PID controller embedded within cells. However, all of these applications have the advantage of guaranteeing a tunable self-regulation of gene expression within the cells with good static and dynamic performance. However, all of these solutions have been designed to work at a single cell level, which means that since there is a lot of circuitry to be implemented within the cells, there is the risk of having a very high metabolic burden on the host, impacting the functionality of the overall circuits, a low modularity because you need to implement everything in the same cell, and if you want to, for example, change the process, you need to change the circuit you embed in the cells, and it's not reconfigurable for the exact same reason. Therefore, we propose an alternative approach that overcomes these limitations by proposing a feedback control architecture that is decentralized, and each of the functional modules that realize the entire feedback loop, so the proportional, the integrative, the derivative controllers, and the process, are relegated within different populations within a microbial consortium. Specifically, we have the, can you see the mouse?
Yes.
Okay, great, sorry. So we have a a process that is hosted in a population that we call targets. And we have the different controllers, so the proportional controller, the integral controller, and the derivative controllers that are embedded within different populations in a microbial consortium. Of course, to make this work, we need also to design some communication mechanism, and we use a quorum sensing mechanism using two orthogonal channels. One that is a channel, there is a molecule produced by the target population that diffuses into the environment and into the controller cells, broadcasting to the entire consortium the state, so the concentration of the protein we want to regulate within the targets. On the other hand, we have another orthogonal channel that basically plays the role of the control action that is produced in different populations, in different amounts, producing a contribution to the overall control action that is basically the molecule that will then diffuse, in the environment from the different populations. And by communicating from the controllers to the targets and from the targets to the controllers, we can close the feedback loop. Of course, we have also the ability of having a tunable response in the target by using a reference signal that is provided to the controllers, and that will be used to set the set point for the target population. The advantages of this approach is that, of course, the modularity is greatly increased, and the metabolic burden associated with each population is reduced, since each functional module is relegated within a different population. Of course, this approach comes also with some limitations and some drawbacks, which are, first of all, that we need to co-culture multiple populations, up to 4 in the full design, which basically needs an extra control over the composition of the consortium. And also, of course, the design is a bit more complicated because other than just designing the targets and the controllers, we also need to design the communication mechanism and ensuring that the outputs of all of these populations are compatible with one another. We started by implementing the simplest controller within this family, which is, of course, the simplest configuration that guarantees a higher performance in terms of regulatory precision and robustness. And it is a multicellular integral control, where we have the controllers or the integral controllers that take as inputs the reference and the output coming from the targets, and output a control signal that then diffuses into the environment and into the targets and regulates the process. The targets themselves then broadcast their state via this Y, which is these other quorum sensing molecules, that then is read by the controllers to change the control action. Basically, for this controller architecture, we proposed an abstract biological implementation that is pictured here, and then that I will explain bit by bit. First of all, we have the controller population and the process that we want to control, which is this XC. XC is a protein that is produced proportionally to the concentration of the quorum sensing molecules coming from the controllers and in the target population. In turn, the targets produce proportionally to the concentration of the molecule that we want to regulate, which is XC, a molecule QX that is here produced proportionally to the concentration of X. This signal, so this QXT, is then received by the controller population, which is built around a titration mechanism figuring 2 molecular species, Z1 and Z2, each produced proportionally to the reference signal and the output signal. And it has been shown in different works that this mechanism here implements an integral control action. The architecture is then completed by the communication architecture, where each molecule, so this Qi, Q, and Qx, diffuse in the environment following a concentration gradient. And of course, in all of the cells, all of the species degrade due to the dilution caused by cell growth and division. This simple model is able to capture the rich dynamics of our multicellular integral controller, and it can be shown that assuming that it can be shown that the equilibrium point is unique, and if it is admissible, which means that all of the quantities, so Z1 at steady state, Z2, Qx, etc., are positive, and if the equilibrium is stable, we can guarantee that at steady states, Xc, so the molecule that we want to control, assumes a value that is proportional to the reference signal Yd. We validated this architecture using BSIM, which is an agent-based simulator specifically designed to simulate bacterial populations, where each cell in the consortium is modeled as a separate agent, implementing a set of gene regulatory networks. In this case, we have two populations, one implementing the controller gene regulatory network and the other implementing the target one. And in this environment, we can also model cell growth and division. We can model the effects related to cell movement, the spatial distribution of cell, as well as the diffusion of chemicals, in this case, the quantum sensing molecules and the reference signal in the environment. As you can see in the video that I'm playing right now, where you can see on the left, cell growing in a microfluidic chambers, the red cells are the controller cells, whereas the green cells that are changing color are the target cells whose color depends on the fluorescence on the level of Hc, which is assumed to be a fluorescent property. And as you can see here, the cell grow and divide in the chamber, and the Xc evolves and goes towards a set point that is here set at 250 and settles at that reference value after a certain transient. So this preliminary in silico validation tells us that this architecture can work. However, as we all know, between theory and numerical simulation and the actual implementation of the circuits, there can be some great discrepancies. As such, we implemented this abstract biological design in a two-strain E. coli consortium. Specifically, we thought of this design that is built around a sigma anti-sigma cognate pairs. Specifically, the controller population is constructed around, as I said, around a sigma factor and its cognate anti-sigma factor. The production of sigma through this double inhibition chain is produced proportionally to the reference sigma, which in this case is IPTG, whereas the anti-sigma factor, which plays the roles of Z2 in the abstract biological design, is produced proportional to this 3OC6HSL molecule, which is the output of our target population, which is to say our Qx. The free available sigma then drives the expression of using this P29N2 promoter of this LASI, which is an enzyme responsible for the production of 3OC12HSL, which in our design plays the role of DQU. These two molecules, so the sigma and anti-sigma, implement that titration reaction that is depicted here in the abstract biological design, and implement, therefore, the integral control action. The signal produced by the controller, so the 3OC12 HSL, diffuses then into the environment and into the target cells, activating through the promoter PLAS the production of both LAX-I, which is an enzyme responsible for the production of the QX, which is to say of 3OC6 HSL, and of a green fluorescent protein, which is used as a reporter to measure the output of the target population via flow cytometry. We validated this biological implementation in vivo using the following pathway. First, we designed the circuit, as I showed you in the previous slide. Then we constructed the plasmid hosting all of this gene regulatory network and transformed them in a suitable equalized strain. Specifically, we used MG1655. Then we investigated the input-output response of controllers and targets by their own by assessing whether the controller and targets could respond to their respective inputs, and tested the communication architecture by basically testing whether the molecules produced by the controllers could be sensed by the targets. And finally, we assessed the architecture performances by gauging the regulation capabilities of the feedback control loop and benchmarking them with an open loop architecture and assessing the robustness of the controller so that we could guarantee both reliability of the architecture and robustness. Let's start with the first step. The first step, as I said, is the characterization of the population to ensure that each circuit can respond to its input. Specifically, we designed an experiment where we set up the cultures of controllers and targets exposed to different levels of IPTG and 3OC6 and 3OC12, grew them for 3 hours in LP, and then measured the fluorescence of each population by flow cytometry after 3 hours. And we saw that the controller population, where we swapped the the LAS-I with a GFP to record actually the fluorescence of the population, could respond to both IPTG and 3OC6 HSL. Specifically, increasing the concentration of IPTG, we could induce an increase in the fluorescence of the controllers, which means an increase in the output that the controllers will be exerting to the target population, whereas the more the concentration of 3OC6 HSLs, which is to say, the more signal we receive as output from the targets, the less the control input will be. Meaning that we can somehow perform a difference between the reference value and the output coming from the targets. Similarly, we tested the response of the targets to the 3OC12 HSL, and we saw that they could respond to increasing concentration of the output of the controllers, which is to increasing concentration of the control signal by increasing the fluorescence of, so the GFP fluorescence that is exhibited by the target's population. Then, as I said, after we characterized the response of targets and controllers on their own, we try to mix them to see whether they can communicate and whether this communication led to a successful regulation of gene expression. To this aim, we also designed another architecture configuration, which is an open loop that we use as a benchmark to highlight even more what are the advantages of having a feedback control loop. Specifically, the open loop benchmark architecture was constructed simply by deleting the anti-sigma plasmid. In this way, basically, we have that the controllers can regulate the expression of GFP in the targets, but their regulation will not be affected at all by the state of the targets. As such, we tested the performance of the architecture both in open loop and in closed loop, using performing a time course experiment where we first set up the cultures of controllers and targets on their own, and then every hour we mix the cultures and incubated them growing, repeating this process for 6 times, and then after 6 hours we measure the fluorescence of all of the co-cultures that will have the same optical density after 6 hours, but will be mixed and that will be mixed cultures of targets and controllers for different times. For example, by recording the performances of the open loop architecture, we can see that after 6 hours of targets and controllers being mixed at different levels of IPT, with different levels of IPTG in the medium, we can see that the regulation at different levels of GFP can happen. However, we can also notice that this regulation is highly variable with respect to the biological replicate that we do, meaning that if we perform the same experiments with 3 micromolar IPTG, for example, 3 different times, we will get different steady state concentration of GFP, meaning different average fluorescences recorded at the flow cytometry. To enhance the performance of this open loop architecture, we can close the loop, and if we do that, we can clearly see that the variability among the biological replicates is greatly increased at the cost of a lower induction or overall induction level of the architecture, because of course we have the 3OC6 that will bring down a bit the signal exerted by the control. So as a sum up message from this slide, we can see that closing the loop, so having a feedback control architecture, we can have a more precise regulation of gene expression. We also tested the performance at steady state of open loop and closed loop architecture by measuring the input-output response at steady state, titrating IPTG over a wider range of concentrations. Specifically, in this experimental setup, we set up the mixed culture from the beginning with different concentration of IPTG. We grew each culture for 6 hours and then measured the fluorescence after 6 hours at the flow cytometry. And as we can see here, basically, both in open loop and in closed loop, we have a first phase where at very low concentration of IPTG, the cells are not responding to the increase in the input signal. And then we have a pseudo-linear increase both in closed loop and in open loop, with a more pronounced saturation in open loop. And then we have an abrupt increase in fluorescence as we go past 8 micromolar IPTG due to the cells starting growing a bit sick. To gauge the linearity of the response of open loop and closed loop architecture, which is quite an important features of feedback systems, we fitted all of these points in open loop and in closed loop with a first order polynomial, so with a line, and then we assessed the performance, the prediction capabilities of the linear model on the actual points. And we saw that both in terms of R squared and in terms of normalized mean square error, the closed loop architecture predicts much better the experimental results with respect to the open loop, meaning that basically the closed loop data points are much better represented by a line than the open loop data, where basically we have this saturation that disrupts a bit the linear behavior. But in any case, we have that both configurations can regulate gene expression with the closed loop having a much more precise regulation and a much more precise regulation and a much more linear response. After that, we also tested the robustness of the consortium, the robustness of the architecture, and we tested this to one of the disturbances that is impossible to avoid when working with the consortium, which is imbalances in the number of controllers and targets that we have in the population. Why is this important? Because if we have, for example, a disproportion of controllers with respect to targets, so we have much more controllers than targets, if we don't have any feedback, then we will be producing a lot more C3OC12, and therefore we will increase the level of GFP expressed by the targets. On the other hand, if we don't have this upper chain here, and we have also just the feedback, basically, we will be producing more C12 and the output of the controller will be reduced. However, if we have both the forward, so the communication from controllers to targets and from targets to controllers, these two effects should balance out. And we tested this by setting up mixed cultures at different inoculation ratios, growing them for 6 hours, and then measuring the fluorescence and the flow cytometry. And we see that by looking at this graph that pictures the normalized steady state fluorescence of the target population with respect to the target percentage at steady state in the consortium, we see that basically the closed loop architecture do not have any influence in terms of fluorescence with respect to the target's percentage, means that no matter what the target, no matter what the composition of the consortium is, the fluorescence at steady state of the target will remain the same, whereas we have a clear correlation, negative correlation between the target percentage in open loop and the normalized fluorescence expressed by the target at steady state. In this graph, all of these dots of different size constitute data points corresponding to the same replicate. So same size is the same biological replicate. Different sizes are different biological replicates. Then the line is a linear regression of all of these blue data points and all of these yellow data points. And the blue line, the blue shaded areas are the confidence interval of the prediction of the result. And if we compute also the slope of these curves together with their confidence intervals, we see that all of the closed loop cluster around 0, meaning that there is, at all of IPTG concentration, there is no influence of the consortium composition on the fluorescence expressed by the target at steady state, whereas in open loop, we have two different cases. For 0 and 3 micromolar IPTG, we have a huge dependence of the target's percentage, so the consortium composition on the target's percentage, whereas even though these confidences are quite large, they encompass 0, meaning that 0 could be a possible slope for these curves. And this is due to the fact that when we increase the concentration of IPTG, the saturation effects due to the saturation of the PLAS promoter are being increasingly important, and therefore slight variations in the concentration of the control input do not impact the fluorescence of targets at steady state. So after all of these, after all of these considerations, we can say that the integral multicellular control architecture works, and it can regulate reliably and robustly the expression of a green fluorescent protein in the target population. But this is just the first step, because we, in this case, designed, built, and tested a distributed integral controllers in E. coli. However, what we want to do is to build the whole PID multicellular control scheme. And to do that, we took the first steps by analyzing both theoretically and numerically the different configuration of the architecture. And in the second part of this presentation, I will show you the PI control architecture and the full PID control architecture. So let's start with a PI control architecture. And let's start by saying that in this case, our control goal is to regulate again a desired protein, in this case XC, to the desired value by bringing the control error to 0 with a low settling time and a low overshoot. And the biological design that we propose to do that is the one that I pictured here in this slide, where we have the process that is hosted in the target population composed by X1 that promotes the production of XC, which is the molecule we want to control. XC in turn produces the output of the target, which is this QXT that diffuses into the environment and then is compared to the reference value YD, both in the proportional controller population and in the integral controller population. The proportional controller population in blue here then produces a control input QUP whose form will be proportional to the control error, and the integral controller will then produce a control input QUI that will be proportional to the integral of the controller. These two contributions to the control inputs will be then summed by diffusing both in the same environment and then diffusing into the target population, closing the feedback loop. Of course, building a mathematical model for all of these architectures starts to be a little bit cumbersome because there are a lot of equations involved, and so the dimensionality of the model will be quite high, and taking out any analytical things out of the very large model becomes very impractical. As such, we made some modeling assumptions to reduce the order of the model and basically, under some assumption, say something about the static and dynamic performance of the model. The assumption that we made are three. The first one is that we are in a situation where cellular population grow and divide at the same rate, so that if the consortium is balanced, so there are the same number of controllers, proportional controllers, integral controllers, and targets, this consortium composition will not change over time. Then we assume that the quorum sensing molecules QX and QU diffuse faster than the aided rates. And finally, we assume that the annihilation process between the species Z1 and Z2 is fast enough, which is a very common assumption when dealing with integral control, with biomolecular integral controllers. Under these assumptions, first of all, we can assume the quorum sensing molecules to be at steady state. So we can assume QUT, so the concentration of the control molecule in the target population, in the controller population, and the integral controller population be the same and equal to some quantity QU bar, whose expression we will see in a second. And same goes with QX, which is the output of the targets, that will be proportional to the concentration of XC, which is what we want to control. The model under these assumptions can be written as a three-dimensional simple dynamical model where we have the evolution of X1, which is the actuating species, whose concentration is driven by this QU bar, which is the control inputs coming from the proportional controller and the integral controller, and the dynamics of the zeta1, which is the difference between Z1 and Z2, which is just the control error. meaning that if I integrate these, I will just get the integral of the control error. And as you can see, my control action QU bar is made of two different contributions. One contribution coming from the proportional controller population, which is a nonlinear function figuring a contribution proportional to the control error. These other bits are to make this contribution always possible, positive, which is admissible. And then we have another contribution, which is proportional to zeta1, which means is proportional to the integral of the control action. Under these assumptions, we can have a null steady state error if the two conditions are met. First, the equilibrium point, the only equilibrium point of the system is admissible, and this can be guaranteed by having beta p, which is the gains of the proportional controller population, be low enough. And how low depends on the parameters of the process. And also, if we have also the integral controller to be upper bounded by this quantity, we can also guarantee this local stability of the equilibrium. We validated these theoretical results by simulating the system, the full system, so the system without any modeling assumption, in the agent-based simulator BSIM, using different controller gains that all respect these assumptions here. And we can see that in all cases, we can regulate the expression of QX, meaning the expression of XC, to a desired value imposed by the reference signal that in this case is depicted as this black dotted line. We validated the nominal performances of this architecture, but we also wanted to see whether the robustness properties that we observe experimentally in vivo are also old in this extended architecture. And to do that, we tested the robustness to two different factors. The first one is cell-to-cell variability, which is always present when dealing with cellular population, which are always heterogeneous. And specifically, what we did is having all cells have each only its set of parameters, and when a cell divides, the daughter cells in the agent-based simulator will have different set of parameters. These parameters will be drawn from a normal distribution centered on the nominal value and with a standard deviation that depends on the coefficient of variation that we vary over the different simulations. And we see that the PI control architecture that we benchmarked against a controller that doesn't have the integral action, which means does not have such precise regulation in steady state, we have that the proportional controller increases the error as we increase the coefficients of variation, whereas the proportional integral controller have an error that stays more or less increases, but very slowly, and has very low values, even if the coefficient of variation that we set is very high. So it's 30% of the nominal values of the parameters. We also tested the robustness of the regulation to the same thing that we tested it against in vivo, which is robustness to consortium composition. In this case, we simulated the architecture varying the integral controller percentage and the target percentage. Note that, of course, the third population will be automatically computed by total number of cells minus target percentage minus integral controller percentage. And so these are all of the possible configurations of the consortium. And we can see that also in presence of very large discrepancies between the number of integral controllers and targets, we can always achieve a very low percentage error at steady state, meaning that the consortium is robust with respect to this factor. Finally, we want to complete this PID multicellular control architecture by adding the last population, which is the derivative control, that embeds a gene regulatory network inspired by chemotaxis that uses two enzymatic reactions to approximate the derivative of QXI, which is the output of the controller, the output of the target population. Under the same modeling assumptions that we use to model the PI architecture, we can see that the model is still three-dimensional, with the control action having now three different contributions. The first two, which are the proportional and integral contribution that we have already seen in the PI configuration, and then a third action that is proportional to this molecule, the concentration of this enzyme A, which is then at steady state proportional to the derivative of the control action, of the control. And in this way, we have a full PID architecture. Of course, in this case, we can guarantee again, when we have the presence of the integral controller and the equilibrium is admissible and it is stable, we can guarantee null steady state error. Whereas with the P and the PD controllers, we can just guarantee bounded steady state error. But in this case, we can also tune the gains to have specific performances in transient. And we do that, so we tune the gains of the closed loop system by using a frequency approach and using the root contours methods, which is based on the computation of the transfer function of XC with respect to the reference of the system linearized around the equilibrium point, Specifically, the roots of this polynomial here are the closed loop poles of the system, and by changing the gain, so the Ki, Kd, and Kp, we can change the poles of the system and so their performances. The root contour method is a method that is based on root locus, and that we basically have. We can set first two gains to 0. In this case, the integral gain and the derivative gain to 0. And so we design a proportional controllers, and we can see that basically starting from the real axis, we have two poles that go along the imaginary axis. And so we have that increasing the proportional gain and not having any derivative gain or derivative action or integral action, we can just increase the elongation of the response without enhancing the settling. But if we fix a proportional action, if we fix a point on this curve, we can derive a family of other curves, which are the closed loop poles of a proportional derivative control. And in this case, by setting the Kp, we set the starting points of the closed loop poles when Kd is equal to 0. And then increasing the Kd, we can move these poles to go towards the real axis and then towards the 0 sin 0. And 1 towards minus infinity. So we can, in this case, reduce the elongation of the response and also decrease the settling time of the system. Finally, setting also the Kd, we can set the proportional gain. So we are on this curve here. And varying the Kd, we can move along this curve here. Fixing Kp and Kd, we can have the starting points of the PID architecture. So we are incrementally building the proportional, then proportional derivative, then the PID architecture. And by increasing the KI, we can have different behavior where we can reduce the elongation and reduce the settling time after a certain bit, and then we have worsening of the performances up until a certain point where we lose also the stability. So using these methods, we can basically tune the gains of the architecture so as to have desired static and dynamic performances. And we validated these over all of the family of PID controllers using agent-based simulators where different gains are tested, all of which respect that the steady-state error must be bounded, the settling time must be contained in 24 hours, and we wish to have a very low elongation of the response. And we can see that starting from a proportional controllers and then adding bit by bit the integral and the derivative action, we can have different performances. Specifically, adding the integral control action, we have a much more precise regulation independently of the controller gains chosen. Whereas adding the derivative control action, we reduce quite a bit the elongation and the settling time of the system. And so we can see that the PID controller, so the full architecture, has an outstanding sobrelongation, so a zero sobrelongation, while having a very contained percentage error and a very precise regulation. So whatever the gains, they always settle to the same points, paying the cost of a slightly higher settling time. Whereas the PID has the best dynamic performances, but also the worst static performances. So depending on what you want to achieve, you could select one over the other. Also, for the full PID control family, we tested the robustness to consortium composition by plotting here the controller's percentage, which is assumed for simplicity to be the same. So we have the same number of proportional, derivative, and integral controller cells, and measuring the steady state error varying the consortium composition. And we can see that when we have the integral controllers, so in the PI and PID configuration, the steady state error always remains bounded and very low. Whereas when we have the P and the PD controllers, after a certain discrepancy in terms of targets and controllers, the error starts increasing a lot. And so if we plot here the areas where the error is bounded over the 10% of the reference, and also we have acceptable transient performances, which is settling time below 1 day, we can see that the P and PD fails after a certain point because the error increases too much, whereas the PI and the PID never fail because of the control error. But if we increase the discrepancies between number of controllers and targets too much, the dynamic performance starts to worsen. And we have the PI, which is the most robust architecture, but very closely followed by the PID, where the derivative, the addition of the derivative control action decreases a bit the robustness of the architectures. So I'm almost done. So in conclusion, we have investigated a multicellular integral controller, and we have seen that it can effectively regulate gene expression in vivo. We added proportional and derivative action to enhance the performances of the controllers developed. And we have tested the performance and robustness of these PI and PID design using realistic agent-based simulators. The open problems that remain open are building the proportional and derivative controllers in E. coli and testing the whole architecture in vivo. And then, of course, one thing that I just mentioned briefly is that we also need to develop methods for the tuning of the consortium composition, because even though I said that this architecture can work well in a wide range of consortium composition, eventually, if I leave them on their own, one population will just outgrow all of the other for the exclusion principle. And so if we want to achieve long-term coexistence of all of the population, we also need to develop methods for tuning long-term the composition of the consortium. And with that I'm done. I want to really thank all of the supervisors that follow me during this journey, which is Mario Di Bernardo, Nigel Savory, Lucia and Claire, and also Vittoria and Davide that were a big part of the second part of the talk. If you want more information about what I've talked during this talk, you can see these preprints about the multicellular integral controllers, and these two works on the control system letters that are on the theoretical and numerical validation of PI and PD control architectures. Thank you very much for your attention, and if you have any question or comments, I'm here and I will be pleased to answer.